My name is Ron Cappelletti. I'm an emeritus faculty member in the physics department, and I want to welcome you all to the public lecture by Venki Ramakrishnan, which is entitled The Quest for the Structure of the Biological Machine That Reads Our Genes. In 1971, if I remember correctly, it was Professor Ernst Breitenberger who was in charge of the committee admitting students into the Ohio University graduate program in physics. Breitenberger had a broad educational expertise, having received doctorates at Vienna and Cambridge, and served as a faculty member in Malaysia, amongst other places. And as such, he had a more than ordinary appreciation for the various kinds of backgrounds from which our applicants from abroad came. Perhaps that was one reason the physics department was open to accepting a 19-year-old student with a 3-year bachelor's degree from Baroda University in India, and perhaps another reason was that Baroda's physics program included study of the esteemed Feynman lectures and the Berkeley physics course. Or perhaps Professor Breitenberger and his committee saw something special about Venki Ramakrishnan and his application materials. In any case, after spending a summer getting up to speed at the University of Illinois at Urbana, by the way, my alma mater, Venki came to Athens in the fall of '71 and entered onto a path that changed his life, and it is fair to say that of Ohio University as well, since here he is. Today, it's only Nobelist. Sir Venkatraman Ramakrishnan, 2009 Nobelist in Chemistry, President of the Venerable Royal Society of London, member of the renowned Medical Research Council Laboratory of Molecular Biology, and Fellow of Trinity College at Cambridge University, winner of many awards and positions too numerous to list here, took his PhD at Ohio University with Professor Tomoyasu Tanaka. By the way, there's a building named after him not far from here, on ferroelectric phase transitions. And a few years ago, Venki, tell you a little story. He said he picked up the paper that was based upon his thesis and he didn't understand a word of it. Besides that, in the 5 years that he was here, approximately 5 years, his conviction grew that he should work in biology, perhaps applying methods from physics, and his life was also changed forever when he chose Vera Rosenberry, whom he met here, to be his wife and partner. You can follow the meandering path of his career and gain real insight into the complex and deeply human nature of how science is really done by reading his excellent memoir, The Gene Machine. And by the way, there are copies in the back in case you hadn't noticed. So perhaps Venki will sign them for you. It is my honor to introduce him and a great pleasure to have Venki back to tell us a real scientific adventure story about the molecular machine that makes proteins and is therefore at the center of life itself. Please welcome Venki Ramakrishnan.
Thank you very much, Ron, for that really nice introduction. I'm really touched that Ron came all the way from Washington to be here today. So I'm going to plunge straight into the talk because I don't want to take up more of your time than necessary. And I want to talk about what the themes in this book are. And the problem is, when young students or the public reads about science, they read a very sanitized version. In hindsight, science is a series of beautiful logical steps, one after the other, and it's so wonderful. And you go away thinking, oh, I could never be like that, or how amazing it all is. And the reality is somewhat different. So what are the 4 themes in this book? The first is the ribosome itself. Now, nobody's heard of the ribosome, unless they're molecular biologists. Certainly most people in the physics department probably don't know what a ribosome is. So, part of the motivation was to tell people about the ribosome. It's this ancient machine that really reads the code, the blueprint of life to make life possible, to make the structure of life possible. The other is my life as an outsider who suddenly found himself in the middle of a high-stakes race. I mean, it is a slightly sad fact that, science is not distributed equitably. It's often distributed in little cliques. If you're in Harvard or Berkeley, you have all sorts of advantages of knowing people, knowing connections, having infrastructure, etc. And I, went to my local state university in India, and then I came here, and then I worked at national labs, which are well known for physics, but less so for biology. And suddenly I found myself in the middle of a high-stakes race for the structure of this machine. And then a third aspect of the book is about the human messiness of science. Scientists are people, and so they have all of the same problems that people have in general, which is they have personalities, they have ambitions, egos, rivalry. And then there's always the question of competition versus collaboration, and also the effect of prizes and what that has on science and their behavior. And finally, for those of you who still have time, there are some lessons that I could think of in hindsight. It's obviously too late for me now, but it doesn't matter. So I'll start off with the science first, which is, it's about genes. My book is called Gene Machine. And no one's heard of the ribosome in the general public, but everyone thinks they know what a gene is. You tell people about genes, they say, oh yeah, I know what genes are. And you say, well, what are genes? They say, oh, they're things that we inherit. They give us our character. You have good genes, bad genes, genes for intelligence, genes for being an athlete, whatever, height, skin color, eye color, etc. And but then you ask them, well, what exactly is a gene? And you might get a little further and say, oh, it's something to do with DNA. But then if you probe them further, they actually don't know what a gene is, okay? So let's talk about genes. First of all, each organism has lots and lots of them. We have about 25,000. And although this says 35,000, that's an overestimate. Now, here's a weed, Arabidopsis thaliana, which is very commonly used in plant sciences. It has 25,000 genes, same number as us. So we're no more complex than a weed, okay? And here's a worm, a soil worm called C. elegans. Several Nobel Prizes have been won by studying C. elegans. And it has only 20,000 genes. It has 20,000 genes, and we only have a few thousand more at best. So we're not more complex in that sense than a worm or a weed. And it should give us some humility. And the reason is that biology does not select for intelligence. It selects for survival, okay? And, whatever helps you survive is what counts. So we have lots of genes. What do genes do? Well, what genes essentially are information on how to make proteins. And why is that important? Because each gene has information to make a particular kind of protein. You could think of a gene as a page in a cookbook to make a particular thing. And proteins go off and do carry out all sorts of functions. They carry out chemical reactions, they give structure, they form your hair, your skin, etc. And if you ask what are proteins in chemical terms, they're long chains of amino acids. So they're polymers, single-chain polymers of amino acids. Amino acids are chemical entities. They have an amino group at one end and an acid group at the other with different side chain groups attached to it. And there are about 20 of them. So you can have different chains which have different permutations of these 20 amino acids. So you can think of a protein as a kind of a sentence where the letters represent the amino acid. So if I represent each amino acid by a one-letter code, then I could represent this protein as a string of letters, okay? Now, one of the miracles of biological chemistry is that when this chain is made, it contains within itself the information on how to fold up into a three-dimensional shape just by chemistry. Now think for a minute how remarkable this is. It would be as if I had several strips of paper. On each strip of paper, I wrote down a different sentence of different lengths, okay? And depending on what I wrote down, the strip of paper would automatically, as soon as I'd finished, fold itself up into its particular characteristic shape. That is how amazing it is. I would call it a miracle, except it's chemistry, okay? So what are some of these shapes? Well, here's something that's like a three-stranded helix. Those of you who wonder what a three-stranded helix is, a traditional woman's braid is a three-stranded helix, okay? And that's exactly what this is. And this is called collagen. It makes up your skin. It makes up your cartilage, your connective tissue, and so on. Without it, you wouldn't be able to stand up, okay? You wouldn't have any form. This protein is called hemoglobin. It contains iron atoms to which oxygen binds in the lungs, and then this molecule, which is in our red blood cells, is carried through the blood to our tissues where the oxygen is released, where they're needed. And then this protein sits in the surface of cells in our eyes, in our retina, and it is what senses light. It's an exquisite photon detector, and it's called rhodopsin. Now, the reason I gave you these three proteins is they have totally different shapes and totally different functions. And now remember, there are thousands of these, okay, each having its own shape and its own function. And every one of them is made somehow by reading instructions in our genes, okay? So how does that work? Well, genes in nearly all organisms, except for some viruses, resides in a molecule called DNA. This is the famous Watson and Crick photograph with their model of DNA taken in the Cavendish lab about 3 miles from where I work. And DNA is a double helix, and unlike the 20 types of amino acids that make up a protein, DNA consists only of 4 types of building blocks, but they're very different building blocks called bases. And the bases have names. They're called A, T, C, and G. And the brilliant insight that Watson had when constructing this model was to realize that A would only pair with a T and C would only pair with a G on opposite sides of the helix, and that an AT pair and a CG pair would have the same shape, so that the helix, no matter what the sequence of bases, in other words, what the sentence of the DNA was, the overall dimensions of DNA would remain about the same. And the other really important thing that came out of it was that if you knew the order of the bases on one of the strands, because of the complementarity, you could figure out the order on the other strand. This meant if the 2 strands separated, each one had the information to make the other strand. And that's how one copy of DNA can be replicated into 2 copies. And that's the basis of all life. You have to have cells that divide into 2, okay? But what was not clear from the DNA structure was how the order of bases in DNA could somehow specify how to make a protein. Because proteins are made of completely different types of polymers, which are chains of amino acids. And so how does that work? Well, a molecule of DNA in us contains thousands of genes. We have 23 chromosomes. So our 25,000 genes are distributed among our 23 chromosomes. Each chromosome is a long molecule of DNA. So you can think of DNA as a library of lots of books, or a cookbook of lots of recipes, if you like. But if you go to the British Library or the Library of Congress and you say, I want to borrow that book, they won't let you, okay? Because that's an archive. They don't want to mess around with it. What they will do is they will provide you a copy of the book, okay? It used to be a microfilm, now it's a digital file. And you can read that file, you can read that copy. And the cell does something very similar. When it needs a particular protein, it makes a copy of the gene for that protein. And that copy is like a copy of one of the strands of DNA, and it's called messenger RNA. It's a slightly different form it's very similar to DNA. One stands for ribonucleic acid, that's RNA. And in DNA, the sugar lacks an oxygen, so it's called deoxyribonucleic acid. That's why it's DNA, okay? But otherwise, they're very, very similar, except one of the bases, a T, is replaced with a very similar base called U. But that's a slightly technical detail. Now, how is this read? Well, if you wanted to read a sequence of bases and convert that to a sequence of amino acids, then how would you do it? Well, if you said each base represents an amino acid, you'd only be able to code for 4 amino acids because there are only 4 bases. But there are 20 amino acids that you have to code for. If you said, okay, I'm going to read bases 2 at a time, well, that wouldn't be enough either because you'd have 4 times 4 combinations, and that would still only get you 16. And so not surprisingly, nature uses a 3-letter code. That is, it'll read 3 of these letters as a unit. And that unit is called a codon because it's a unit of the code, of the genetic code. Now, it turns out that amino acids can't recognize DNA directly. They have to be recognized in some indirect way. And Francis Crick, who was one of the people who solved the structure of DNA, said there must be an adapter molecule, which on the one hand might base pair with 3 bases at one end, just like the bases pair across the strands of a DNA, and at the other end it might bring an amino acid along. And it turned out those adapter molecules were discovered, and they're called transfer RNA molecules. They also turn out to be RNA. So remember, you've got a messenger RNA, and now you've got a different kind of RNA called a transfer RNA. And there'll be a third kind of RNA, and I'll get to that later. And then I'll tell you why there's so much RNA in the world. And so it brings along an amino acid and reads the code at the other end. And then a second transfer RNA, and they're called transfer because they're transferring the amino acid to the growing protein chain. So a second tRNA will read the next codon, and so on. And of course, this doesn't happen magically by itself. In fact, while molecular biologists were figuring all this out, it turned out that cell biologists were looking to see where are newly made proteins inside cells. And they found they localized in these little blobs on an organelle called the endoplasmic reticulum. And when they purified these blobs, they all turn out to be particles of about the same size. And if you look very closely at each of these particles, you see a little divider. It's because each of those particles consists of 2 parts, a large part and a small part. And so they were called large and small subunits. Now, when the cell biologists were breaking open cells and to purify these particles were found in something called the microsome fraction. And then when they analyzed them, they found they were made up of both RNA and protein. So they were called the ribonucleoprotein particles of the microsomal fraction, okay? And, imagine if you're at a conference and every time you wanted to refer to that, you had to say that whole long phrase, okay? So somebody named Howard Dinsis, who's at Johns Hopkins, said, look, why don't we just call it the ribosome? And that's the word that's stuck ever since. Now, people went on to analyze the ribosome. So when biochemists discover something new, the first thing they do is break it apart and see what it's all made of. It would be like if you're a kid and you want to understand how a radio works, the first thing you do is just take a screwdriver and take it all apart, okay? Now, putting it back together is a much harder problem, and that's true for biochemists too. Putting back the ribosome together took about 15 years after it was discovered. But anyway, they found out that it was made of many proteins, about 50 proteins in bacteria, and it's made of 2/3 of it is made of RNA, consisting of these 3 long molecules. And then these go up to make the 2 subunits, which then make up the whole ribosome. And this is an early electron microscope image of what a ribosome would look like. So how would a ribosome work? Well, a lot of very clever biochemistry. Before any structures, biochemists had figured out quite a lot. The thing they figured out was that the small subunit is what binds the genetic message, and that the ribosome has 3 slots for those little adapter molecules, those transfer RNA molecules that bring in the amino acid. And somehow the ribosome figures out where to start on the genetic message. It knows that this stuff upstream is not real message, and this is where it has to start. Brings in the first amino acid. Then a second tRNA comes in. Now, there are lots of tRNAs in the cell, and they can come in at random. And if they're not right, in other words, if they don't match the code, they're rejected. But if they match the code, the ribosome joins the first 2 amino acids. And then the whole thing has to move. And then once it moves, the first tRNA is kicked out. The new tRNA now becomes the old tRNA, but now notice it has 2 amino acids attached. And then a third tRNA comes in, and if it's the right one, it's accepted, and then it's joined. So you can see what's happening. The ribosome is only choosing the tRNAs that correspond to the code on the message, and those tRNAs have the right amino acids attached to them. So when it's stitching together the amino acids, they're in the order specified by the code on the gene. Okay, so that is the beauty, and that's why it's called translation, because it's translating from the language of genes to the language of proteins. Okay, so all of this was known when I entered the field. Okay, so I'm going to switch now to my personal story, because the book is also written as a kind of interweaving of science and personal stuff. But I have to tell you, it's not an autobiography. I mean, Ron correctly described it as a memoir. It's about the ribosome, and I use my story as a narrative. So, as Ron mentioned, I took off from India, from Baroda, India, to Athens, Ohio, at the age of 19 to get a PhD in physics. And I spent the first 5 years of my life in the US here, and this is Clippinger Labs. This is actually a fake picture. It's from 2008, because there used to be railroad tracks here. Okay, and if you didn't time your entry correctly, you'd be stuck for about 15 minutes waiting for a long freight train to go by. Anyway, so the first year, 2 years, you had to take courses, and you had a comprehensive exam, and all of that was perfectly okay. I did just fine. And then, I started realizing I had a problem. And so what was the problem? Well, the first thing is, the problems in physics are very hard, okay? And what do I mean by that? I don't mean exam problems, I mean that it's really hard to make real headway in physics, because it's a very mature field. It's several hundred years old. And, there had been very few breakthroughs and by breakthroughs, I mean really things that fundamentally changed our understanding of something in the previous few decades. And, I found it quite hard going, and I found that if I stayed on in physics, this is what I would end up doing. I would be doing a lot of boring calculations that didn't improve understanding, okay? I might be able to publish the odd paper, but, it wouldn't really. And let's be honest, I have a colleague in Trinity College, and we talked about high-temperature superconductivity, which was discovered in the 80s, I believe. And, there's still no good theory, but there are thousands of papers on it, okay? So, that's the reality. So then, what about biology? So biology, I used to read Scientific American, and I still do. And almost every issue of Scientific American reported a major breakthrough in biology. In the 70s, that's when DNA sequencing was invented. That's when the first structures of biological molecules were being discovered. The idea that the cell has a membrane, and how the immune system works. All the stuff was coming out, okay? And I knew that many physicists had made this transition before. For example, Max Delbruck and Max Perutz, who founded the lab where I work, and Francis Crick, and so on. So I thought, well, this is a path that has been taken by other physicists. But the problem was that I thought of doing a postdoc, but then I realized I don't actually know any biology, And so, what would I do? So I actually applied for several graduate programs. And one person at Caltech wrote me a scathing letter saying, we're not going to ever accept a guy like you. And two of my students, I'm happy to say, are faculty members at Caltech now. So anyway, but the other thing was that Yale University, the chair wrote me a letter saying, look, we can't take you because you already have a PhD, but I'm going to pass your CV around to faculty members. Maybe somebody will offer you a postdoc. And two people wrote to me offering me a postdoc. One was Tom Steitz, with whom I ended up sharing the Nobel Prize. Another was a guy named Don Engelman. And I turned them both down, saying I didn't know any biology, I wasn't ready for a postdoc, and I wanted to go to grad school. So I went off. My son was born in O'Blennis Hospital, not far from here. And 6 weeks later, or no, actually 4 weeks later, we packed up all our stuff and I drove with a friend across the country from Athens to San Diego, where I went to University of California in San Diego for graduate school. And I spent 2 years of my life there. And the first week, there's a kind of survey lectures by faculty for incoming graduate students. And I sat through them and realized I didn't understand a single word of what they were saying. It was all full of jargon. And so I said, well, this is not going to work unless I take a step even further back. So I went to my graduate advisor and I said, look, I need to take some undergraduate courses. So I took undergraduate cell biology and genetics and biochemistry in my first year at UCSD. And it's only then that I started doing research in biology in my second year. So the lesson is don't worry about how far back you have to go. You have to go as far back as you need to learn something, okay? And anyway, by the end of the second year, I realized I'd acquired a background in biology. I was doing research. I knew how to pipette and all those things and run gels. And so I thought, well, do I really need another 4 or 5 years of this to get a second piece of paper? And around that time, an article in Scientific American was written on using neutron scattering to do the structure of the ribosome. Well, by that time, I knew what a ribosome was, first of all. And I thought, oh, with my background in physics, maybe I can learn neutron scattering. That might not be so hard for me. And I noticed one of the authors was the guy who had offered me a postdoc. So I wrote to him, and I said, you offered me a postdoc when I didn't know a damn thing. And now I actually know something, so are you still interested? And he wrote me back and said, yes, and his colleague Peter Moore would come and talk to me because he was coming to a meeting in San Diego. So I met Peter in San Diego. Peter Moore was the ribosome end of that collaboration, and he got to work on ribosomes because he was a student of Jim Watson's. And Jim Watson pioneered a lot of ribosome research in the 50s and 60s. So we, again, packed up and moved across the country to New Haven, Connecticut, where I got my introduction to ribosomes under this man, Peter Moore, who's been a lifelong mentor and friend. And what we were doing was by deuterating individual proteins and putting them back together in the ribosome, we were measuring distances between ribosomal proteins. And by measuring all possible distances, you can get like a three-dimensional arrangement of the proteins in the ribosome. Now, the problem is the ribosome, first of all, it doesn't tell you anything about what these proteins look like. You just know roughly where the center of mass or where they're located. That's why they're shown as spherical balls, And so when all was said and done, it didn't really get us much closer to understanding how a ribosome worked, okay? So what's the problem? Well, imagine if you're a Martian hovering above the Earth and you watch these little things go along in straight lines and occasionally turn at right angles unless you're in parts of Athens when it sort of makes these zigzags. So, you think, oh, okay, that's something happening here. And then you observe closely and you realize it's taking in gasoline and emitting carbon dioxide and water and some pollution. That gives you, and heat. So then you think, oh, well, this is some machine that uses energy. But you wouldn't understand how it worked. If you got a closer look at it, you'd say, oh, well, this is made of wheels and, something happens to turn the wheels. You get a closer look, you'd understand how it was put together. But really, when you have the details and there are pistons in an engine, and the pistons are forced when there's a combustion, due to fuel explosion and so on, and then there's a crankshaft that converts that motion to the wheels and so on. Then you'd start understanding how a car works. And it's been the same ever since throughout science. You've got to be able to see things to get to that next level of understanding. That's been true in astronomy, starting all the way with Galileo and his telescopes. It's been true in biology. It was because with microscopes you could see microbes and cells that we understood that the cell is the unit of life. So it's been true for everything. It's true for molecules too. Even starting with sodium chloride, people didn't understand how sodium chloride, what the structure was, until Bragg solved its crystal structure and said, there's no sodium chloride molecule. It's a bunch of ions, sodium and chloride ions. So we know if you want to see a small object, like a cell, what would you do? You take a microscope and magnify it and look at it. And the way that works is you'd stick a lens in front of the object, and the lens would collect scattered rays and recombine them to form an image. And in fact, all of you are doing that right now with the image from the screen. The scattered rays from the screen are entering the lens in your eye. The lens is recombining those rays to form an image in your retina. And depending on the relative distance, the image can be smaller, or with a telescope, it's much smaller, and with a microscope, the image is much larger. So that's how you would magnify an image and look at it. But as physicists all know, there's a law which says that you can't resolve two objects. You can't see two objects as distinct if they're closer together than about half the wavelength of the light you're using. That's a famous law by Ernst Abbe from the 19th century. And people already knew that the distance between atoms was thousands of times smaller than the wavelength of light that you can use. So you would think that you would never be able to see molecules. But in 1912, a graduate student named Bragg figured out a way for how to use a technique called X-ray crystallography to be able to determine the structure of molecules. And he became the youngest ever Nobel Prize winner at the age of 25, and he's still the youngest science Nobel laureate. So what you do is you form crystals. What crystals are regular three-dimensional stacks of molecules. And if you stack up a bunch of marbles or oranges or whatever, that would be like a crystal. And you then take the crystal and you hit it with a beam of X-rays, and you collect the scattered rays. Now remember, the scattered rays contain the information to make the image. All the lens is doing is recombining them. The lens is not actually providing the information. The information is there in the scattered rays. And so if you were able to collect these scattered rays, and this is what a pattern would look like, then if you were somehow able to mathematically recombine those scattered rays in a computer, you would be able to regenerate an image of the object. And that is really fundamentally how crystallography works. I go into a bit more detail in the book, but to get beyond that, you would have to really get into some of the mathematics of it. But basically, think of it as a way of, doing what a lens does, but in a computer, and using X-rays instead of light, okay? So I was at Brookhaven National Lab, and I was doing neutron scattering to look at biological structures. And I have to tell you, neutron scattering is a great technique in physics. Ron has used it a lot. But it's a pretty limited technique in biology. It's a very niche peripheral technique in biology. And I realized I wasn't going to be able to solve anything interesting about the ribosome or anything else with that. So when it came time for tenure, they asked me, what will you do if we give you tenure? I said, well, the first thing I'd do is stop what I'm doing and go away on sabbatical. And they were a little shocked. And usually, you give them all these grand plans of, I'm going to do this, that, and the other, and not stop what I'm doing. And so, but then they thought about it, and they said, actually, he's right. And so they gave me tenure, and I went off on a Guggenheim Fellowship to work with Aaron Klug, he died last year. He was, at the time, he was a towering figure in my field. He was a Nobel laureate. He went on to become president of the Royal Society. So when I wrote to him, I had no hopes that he would actually reply. But fortunately, he replied and not only encouraged me, but helped me get a fellowship to go to this famous lab, which I'll talk about later. And I learned crystallography in my sabbatical year in England. I came back to Brookhaven on Long Island, and then I collaborated with a friend of mine who, oddly enough, was from England. And together we started looking at little pieces of the ribosome, each of the proteins that make up the ribosome, painstakingly trying to get crystals of each of them and solving their structures. And so I call this the turning balls into ribbons phase, because now it's no longer a ball, it actually has a structure which you represent as this ribbon diagram to show this sort of fold. But this too, it was interesting at first, but it became a big stamp collecting exercise, because when you look at it, it'd be like if you had no idea what a car looks like, and you go to an automobile parts store and you start examining little parts like a spark plug or fuel injector. You have no idea what it does or where it belongs in a car. And you say, oh, interesting, that's what a spark plug looks like. Well, that's not going to help you understand how a car works. So how would that ever happen? Well, the ribosome was much larger than things that had been crystallized around 1980. Up to that time, the largest molecule that had been crystallized had a molecular weight of about 350,000. And the ribosome was 2.5 million Daltons, okay, and has 500,000 atoms. And things changed when an Israeli scientist, Ada Yonat, went to Heinz Gunther Wittmann to do a kind of, maybe a sort of visiting fellowship. And there's a very funny account of how she happened to end up there, because Wittmann had actually tried to get 2 other people interested in this project before Ada showed up. Anyway, the 2 of them collaborated, and they got crystals of the large subunit, which is the part where the amino acids are joined together of the ribosome. And then another woman, Maria Garber, who was heading up a group in Pushchino, a science town outside of Moscow in Russia. And she and her group got crystals of the small subunit of the ribosome and the entire ribosome. So now you had crystals of both the small subunit and the large subunit of the ribosome. And this was 1980 to about 1985 or so. So you fast forward 10 years. By this time, I was come back from my sabbatical. I was getting tired of stamp collecting, and I had some ideas from my sabbatical on how it might be possible to tackle something as large as the whole ribosome in terms of getting enough signal in the diffraction experiment. And so what was the state of play? 15 years after the first crystals, there was still no really credible structure, not even low-resolution structure from the crystals of the ribosome. And I talk in the book about a couple of really important meetings in the mid-90s that really told many of us in the field that other people better get into the action if they wanted to see the field move. And at the time, there were very good 50s crystals. So the first crystals weren't good enough. And I was talking in the book about what's not good enough means. It means the molecules are not all exactly arranged regularly in the crystal, but they have slightly different orientations. So the overall image is sort of blurred out and you don't see the details. But so you need good quality crystals, and you can tell that by how far out the spots go when you hit it with X-rays. Anyway, Ada Yonat, after about 10 years, had found good crystals of the 50s, the large subunit, but still no progress. Many years later, no progress on a structure. So I didn't want, and then I went to a meeting in Seattle, and I learned that my former mentor, Peter Moore, was collaborating with Tom Stites, his colleague, and they were going to take Ada Yonat's 50s crystals and figure out how to solve it, okay? Because they've clearly, everybody felt she was stuck, and so they thought, okay, let's see if we have different ideas and go with that. And I didn't want to do that. I thought, well, because there used to be an old tradition that, if somebody's worked for a long time on a particular project, you should maybe let them get on with it. Anyway, but I said there are no good crystals of the small subunit or the entire ribosome, and so maybe I should, work on those. And I thought the whole ribosome was too complicated and should come later. So I decided to tackle the small subunit, and by this time I'd left Brookhaven, and for a brief period, I was actually a professor. It's the only time in my life that I've been a professor. I was at the University of Utah for 3.5 years. And this was a picture of my lab, and I persuaded 2 graduate students, Bill Clemens here, and let me just, sorry. So this is Bill Clemens, and this is John McCutcheon. So these were 2 first-year graduate students, and I persuaded them that working on the structure of the small subunit, which is about 150,000 Daltons in molecular weight, was a good thesis project for a PhD. And despite the fact that this large, well-funded Max Planck group in Germany had not made progress on it for 15 years, but it was nevertheless a good PhD project. And since they knew nothing about crystallography or the ribosome, they both said, yeah, sounds good. So sometimes it's dangerous to know too much, because And then this is Brian Wimberly, who was absolutely crucial for building the final structure because of his knowledge of RNA. But he was on his second postdoc, and he knew too much, so he didn't want to join it right away. But as soon as things were working, or looked like they might work, he was happy to join. And this is me in my Bin Laden days. And notice I'm the only one wearing lab gloves. So when this picture was taken, I was the only one who was actually working in the lab. So anyway, so we started working, and we started trying to get crystals of the small subunit. And then I went to a meeting in Sweden, and when I was at the meeting, the Yale group reported progress on the large subunit. And Ada, it turned out, abandoned the large subunit. She showed data whereby she claimed it was unworkable, which turned out to be wrong because the Yale group actually ended up solving it. But she then showed pictures showing that she now had crystals of the small subunit that somehow she had stabilized and were now diffracting better. So instead of the Yale group and Ada sort of duking it out and my having a quiet little niche for myself, I found I was going to be in a head-to-head contest with this extremely well-funded group that had been doing it for a long time, and I had my 2 first-year graduate students, okay, in Utah. So I went back. I had this long, interminable plane trip back from Sweden to Utah, and I was just thinking of giving up the whole thing altogether. But then I thought, look, crystals have been around for a long time, and nobody's yet figured out how to solve it, and I have ideas for how to solve it. So just because someone else has good crystals of the 30S now doesn't mean I should give up. And I also thought, this is the most important problem in this field, and one of the most important problems in biology, how a gene is read to make proteins. And so I thought, if I don't do this and I watch other people do it, I'll just be kicking myself. But if I try and fail, that's life. And then when I was in Utah, Harry Noller, who's a very well-known biochemist at UC Santa Cruz, suddenly I found out he was working on the ribosome. And I thought, how does this guy who doesn't know any crystallography suddenly start doing crystallography of the ribosome? Well, it turned out he had hired two of the Russians who had crystallized the ribosome. And then he had hired this hotshot crystallographer, Jamie Cate, who's now a professor at Berkeley, to be his postdoc. So he had assembled a crack team to work on the entire ribosome. So suddenly, my little niche became a four-way race, okay? Now, what do you not do when you're in the middle of a hugely competitive race? What you don't do is move to a different continent, okay? But that's exactly what I did, okay? And so that was another crazy thing. Why would I do that? Well, here's the problem. I wanted to work on whole ribosome structure, but I had no idea how long it would take. Remember, crystals have been around for 15 years, well-funded groups. Yes, the Yale group was making progress, but it doesn't mean they would get to atomic resolution. Nobody, really knew. This was beyond the edge of what crystallography had done. So I thought, if I stick around in Utah, I have an NIH grant, and if I spend the NIH grant to do this, and at the end of 3 years I haven't made my breakthrough, I'm going to, lose my grant. And if I put in a separate grant, I can just imagine what a panel in Bethesda would say when this guy's grant shows up saying he has some ideas for how to solve the ribosome, and but he doesn't have any good crystals yet, but he's going to give it a shot. They would just all laugh, and toss it in the bin. Okay, so that was the other problem. So I wanted stable funding, and the other thing I wanted to go to a place which had real experts in the technique. So here's the difference. I've used the car analogy a lot. This is a different car analogy. It's no problem if you want to go home from this lecture in your car. You're just going to drive home. It doesn't mean you have to understand how an internal combustion engine works and how a car works or anything, okay? You just have to know how to drive it. But if you're going to take a car across the Sahara Desert, then you better know all the ins and outs of how a car works, because it's likely to break down or you're going to be pushing it to its limits, okay? So I wanted to be a place where people really understood the nitty-gritty of crystallography. And then the LMB, from my sabbatical, I had really liked it because it's a very collegial place and it's a complete lack of hierarchy. We have a $5 million microscope. If you're a first-year graduate student, or if you're the lab, Richard Henderson, who won the Nobel Prize for electron microscopy, it doesn't matter. If you sign in first, you get to use it first, okay? And there's no faculty club or anything. There's a cafeteria. Everybody sits, whether they're a cleaner or the lab director. They all find a place and they sit next to each other. So, and young scientists are given independence very early. They're given some lab space and said, go do what you want. And it's also an institute with a tradition of tackling very hard problems. So Fred Sanger, who has 2 Nobel Prizes, has what's called an H factor of 18. You probably wouldn't get tenure at most places, based on that H factor. And the last 2 years, the lab won chemistry Nobel Prizes to Richard Henderson and Greg Winter. If you ask when did they start their work that got them their Nobel Prize, it was 25 years ago, okay? So they have this long vision that if it's a hard problem but you're making progress, they don't pull the rug out from under you. So those were all the reasons. So I persuaded Vera, who had been, I forgot to mention that the only really notable thing I did when I was in Ohio, apart from passing my comprehensive exams, was to meet and marry my wife, okay? And so she'd been following me around all over the country, and I told her, look, Vera, I really would rather take a 40% pay cut and move to England. And luckily, she didn't say, what are you, crazy? She said, okay. But she said, this is the last time we're moving. So, and so now, you know, if anybody's interested in me, she always said, no, I'm not moving. You're going alone if you're going. So we ended up going to Britain. And then we had this mad scramble. And I won't bore you with all the technical details. I thought we'd almost blown it after moving to England. And I had to assemble a completely new team there. But two of the people from Utah, Brian Wimberly and Bill Clemens, came with me, luckily. And this is Brian with stereo glasses trying to fit, you know, a 3D image, a molecule into a 3D image in front of a stereo graphics computer. Of course, now, you know, you can get, any kid can get this for video games at home. But this used to cost, you know, thousands of dollars, I mean, tens of thousands of dollars in those days. And the other thing we needed were synchrotrons. These are very powerful sources of X-rays, which are basically electron accelerators. And so we used to have to go to, you know, wherever we could get beam time. And these are some of the synchrotrons we used. And this just shows you, you know, so I had to, because of the way I was collecting data, I wanted my crystals to be perfectly aligned around a symmetry axis of the crystal so that the pattern would be symmetric, because the small difference between the left and right side would give me information I needed. And I explained that in the book. So there's only one place that had both the high intensity and this ability to align crystals. And that was at the APS in Argonne, outside Chicago. And it took me 6 months to get beam time. And it only happened because my professor at Yale had a colleague who was a professor at Yale who was on the committee for that beam line. And he put in a word for me, you know, and it's only when he put in a word for me that I got beam time 6 months after I asked for it. And a week after he interceded on my behalf, he died of a heart attack. And, you know, it shows you some of the real vagaries of science. You know, if you're outside this circle of knowing people, you'd be completely cut out, you know? And it's because I knew people who knew people that this was all possible. And even so, you know, if I'd waited a week or two, you know, I might never have been able to have this guy intercede on my behalf. So you know, I talk a little bit about what that felt like. Anyway, this was the beam line in Chicago, which we used, and the X-rays come in here and they hit the crystal and they collect the data. Now, when you do the experiment, it doesn't tell you the structure. What it gives you is a three-dimensional image of the molecule, because biological molecules rarely have the crystals that have such a good resolution that atoms will show up as little spheres, okay? Rather, you see a sort of more blobby but still detailed enough structure. So how do you get an atomic structure from this sort of image? It's a bit like fitting in a large three-dimensional jigsaw puzzle. And the way you would do that is just with a jigsaw puzzle, you know, but there's some differences. It's three-dimensional rather than two. And of course, you don't have the answer on the cover of the box. But otherwise, it's like a jigsaw puzzle in that you look for patterns that you can recognize and build those first, and then save the hard parts like the sky or grass or something for the very end. And so you do exactly that here also. So you would zoom in, and you can see that there are two sort of strands that seem to be kind of interwoven around each other, and you can see little bumps and so on. And so if you know what you're doing, then you realize that those two strands are really strands of RNA that are interwoven as a double helix, because RNA can also form double helices. So suddenly, you've taken a piece of the image and you've interpreted it in chemical terms in molecular terms. And so there you have a piece of the molecule suddenly. And now you have to do this for the entire image. And so you sort of keep on building until you have the whole thing built. And when you have no more image left to interpret, then you have the atomic structure of the molecule. And doing exactly that, just as we solved the small subunit, the Yale group solved the large subunit of the ribosome. And they both came out within about a month of each other in the year 2000. And one of the things that came out of it is the place where the amino acids are joined together to start making the protein is in this pocket here. And the pocket is made entirely of RNA. Even though the ribosome is made of both proteins and RNA, the pocket is made only of RNA. And similarly, the part which binds the genetic message and where the message is read is almost entirely RNA. Why is this interesting? It's because there was a long-standing chicken or egg puzzle. The ribosome is what makes proteins. But if the ribosome itself is made of both RNA and proteins, how did the ribosome even come into being? Okay? Well, Francis Crick had the idea that maybe early ribosomes were made up entirely of RNA. At the time, it wasn't known that RNA could also carry out chemical reactions. That was discovered later, for which Tom Cech and Sidney Altman won the Nobel Prize. So what we now believe is that life began as a world dominated by RNA, because RNA can carry out chemical reactions, but it can also carry information, like DNA. And then it was used to make proteins, and then eventually proteins took over a lot of the chemical reaction and other roles that originally might have been played only by RNA. And DNA probably may have come later. And so RNA probably is the oldest sort of information molecule in the world. And so the structure shows that. And then 6 years later, we solved the structure of the entire ribosome. And what these structures have done is that if you solve the structure of the ribosome, you can now solve the structure of antibiotics bound to the ribosome. And so we were able to solve lots of different antibiotics bound to the ribosome. And drug companies are now trying to use these structures to try and design better antibiotics, which might have fewer side effects and so on. Now, for the last few slides, I want to talk a little bit about the human side. One is that scientists are neither Spocks nor saints, okay? And so we're motivated by lots of things. We're motivated by curiosity, by ambition, by desire to succeed and be first. We also have egos, we have insecurity and anxiety, we have rivalry and competition. And we're motivated to a large extent by self-interest. So we will collaborate or compete depending on what's in our self-interests. For some things, competition makes sense. For other things, collaboration makes sense. But just like humans are complicated, you can't dismiss humans as just purely selfish creatures, because altruism is also part of humanity. It's a part of our nature. And many people also help me. They give me advice. They give me specialized reagents that you couldn't buy, that they had the expertise to make, technical help. And they all did this without any expectation. They didn't ask for money. They didn't ask for co-authorship, nothing. They just gave it because they felt they were helping a fellow scientist with an interesting problem. So you see both the good and the bad side of humanity in science. Then I talk a lot about, there's a chapter called The Politics of Recognition, about the corrupting influence of prizes. It starts early, even in school. You start getting, first prize, second prize, math prize, etc. Then you start getting early career awards. And then you start getting, people hanker to be elected to their National Academy or in Britain, the Royal Society. And then, even if they do that, they're still not satisfied. Then they want bigger prizes. And, many of these prizes, like the Nobel, have a limit of the number of 3. And, science today, maybe it was okay in 1901 when people were relatively isolated. But today, science is, just having a germ of an idea, it spreads. You mention it at a meeting and it just immediately spreads. Lots of people start working on it. So you don't know if that original idea was the really important thing or some later thing was very important. It's very, very hard. Now, another thing is that the public thinks of Nobel laureates as some sort of geniuses, because, in the early days of the Nobel, coincided with, development of quantum mechanics and relativity and the, genetics and modern chemistry and so on. So many of the early prize winners were really, geniuses. But by and large, it's not given for being not even given for being a good scientist. It's really given for an important discovery or making an important advance, okay? And in my book, I say it's a bit like Shakespeare described it. Some are born great, some achieve greatness, and some have greatness thrust upon them, So you could be in the right place at the right time, or you could stumble onto things, and you could get a Nobel. And I talk about, how people behave when the stakes are high. And, there is a disease among high-profile scientists. There are two diseases. One is called pre-Nobelitis, which is before they've won a Nobel, but they're sort of hoping they'll get one, and, and they have this anxiety every October and so on. And then there's a post-Nobelitis where suddenly, they haven't done any important work for 20 years, and they get a Nobel Prize, and suddenly everybody thinks they're a genius, and they go around the world, their average altitude, instead of becoming 0 feet above sea level, is now about 30,000 feet above sea level. And, they go around pontificating about all sorts of things. So, that's part of it. And of course, it affected the ribosome. As soon as these structures came out, people said, this could win a Nobel someday. And it starts warping your psychology, And so there are conferences all over the world, and there's all this jockeying, people trying to talk up their work and talk down other people's work and so on. And I could see this in all sorts of meetings. And the funny thing was that from 2000 to about 2005 or so, or actually even later, suddenly there were meetings in Sweden that I kept getting invited to, okay? And suddenly the Swedes had become interested in ribosomes, okay? And it was almost like auditioning. And, I saw how people behaved at these meetings too when they saw that members of the Nobel Committee were in the audience. And at one of these meetings in 2004, there's always a banquet on the last night. And the only Swedish speaker at this meeting was a very distinguished ribosome biochemist. He and I had been good friends, but we didn't agree on how the ribosome accurately reads the code. And so he came up after the banquet dinner and started just tearing into me about, my talk and my work and so on. And then at some point I got irritated and started fighting back. And then a colleague of his had to separate us. And then about 3 months later, I found out the guy had been appointed to the Nobel Committee in Chemistry. So I said, well, there go my chances. And so I basically declined all invitations to Sweden because I said, well, I'm not going to be in the running anyway, and why the hell should I go there and eat bad vegetarian food, So I didn't go there from 2004 to 2009 except for one talk which a friend of mine had asked me to give. So I'd completely given up. So one morning in October of 2009, I came to work late because I had a flat tire. I was irritated. I got this phone call saying, please hold the line. This is an important call from the Swedish Academy of Sciences. I thought it was more likely to be a prank call by one of my friends. And I told the guy, you have a pretty good Swedish accent, but I really don't believe you. And then the guy, then I said, well, if it's true, then I want you to put this, ribosome colleague of mine on the line. And this colleague of mine, Måns Ehrenberg, came on the line and said, Venki, it is true, and you did deserve it, but I'm not going to say it ever again. So that was that. So who would have thought, okay? I mean, I went from, from Baroda, India to Athens, Ohio, from physics to, neutron scattering and stuff like that, that I would end up in this place. Now, this is a long table, and I just wanted to show you an interesting thing here. So this is the King of Sweden. Just above the above the red dot is the King of Sweden. This is Ada Jonat, with whom I shared it on next. That's me. That is the Crown Princess Victoria, and that's Tom Steitz. And my wife Vera is busy talking to the German Vice Chancellor over there. Now, every year, there are 3 science prizes given, physics, chemistry, physiology, medicine. So there are about 9 Nobel laureates. And then there's, of course, economics, and there's literature. But clearly, the newspapers realized that that year, it's the ribosome that was really the important thing. And the others were okay, but not that great. And how do we know this? Because the next day on the newspapers, this is what they put on their front pages. There's Tom Steitz and me, 2 ribosome guys on the front page. And of course, the fact that Princess Victoria was in between us had nothing to do with their choice of photograph. Okay. So for my last slide, what can I say? Why did my career work out? I could easily have fallen off the map. After my postdoc, I applied for 50 faculty jobs. I got 0 interviews, okay? And I had to take this job in Oak Ridge where they didn't deliver what they said they would, and then I had to try and get out of there and so on. So the thing is, you've got to keep your options open. If you decide to quit, then you're not playing the game. And if you're not playing the game, you can't get anywhere. And then the other thing I learned is you should never be afraid to ask for help. I learned that from my sabbatical when all these famous scientists at the MRC lab would ask what seemed to be really ridiculous questions, very naive questions. And I realized they didn't mind that they were ignorant. Ignorance is not the same as stupidity, okay? And then you've got to talk to people. Many of the ideas I got were from random conversations, but you can't talk to people all the time. Otherwise, there'll be groupthink. You won't be able to be creative and do that. Another is imposter syndrome. You go to a place and you think, when I went to become a biologist, I thought, oh my God, what am I doing here? This is crazy. Or when I went to Yale as a postdoc, I thought, I don't really belong here. And so everybody has imposter syndrome, unless you're a psychopath, then you probably don't have it. And one thing that if you're a minority of some sort in science, then you find, for example, if you're a woman or an ethnic minority or a geographical minority, you could be from rural, small town, and you're suddenly in the Northeast or somewhere, and you think, oh, I don't really belong here. Don't worry about it. Everybody has imposter syndrome. It's just some people are better at faking the fact that they have it. So that's the thing to remember. Just follow your interest and carry on. And then science doesn't depend on being a genius. It depends on a lot of things. One is having money. Even if you're a theorist, you need a salary. And you need skill, which means you should try to get the best training you can. You also need patience. If you're working on something hard, you can't just give up at the first difficulty. And all scientists in hindsight, not all scientists, but many scientists in hindsight seem to forget the element of luck, okay? I had all sorts of random encounters that, set me on this path. And if I forget that, it's not all my doing, I helped, but, the luck plays a role as well. So I want to finish by showing you this movie, which is how a ribosome, it's a movie of how the ribosome works, okay? So there's a small subunit, and that long snaky molecule is the messenger RNA that contains the genetic information. And in blue is that first tRNA molecule, bringing in the first amino acid. And those other little blobs are proteins that help the ribosome find the right starting point. And now notice the large subunit is joined. And so when these factors leave, the ribosome is ready to start what's called elongation, which is adding amino acids to the growing protein chain. And so each amino acid is actually brought in by a tRNA molecule, but the tRNA molecule is attached to another protein, which is a kind of factor. And so it delivers the amino acids through the tRNA to the ribosome. And you can see how the tRNA then swings in to join the blue one. And at the ends, the two amino acids are being joined. You can't see that because it's in a pocket. And then the whole assembly has to move, and that happens in 2 steps. First with respect to the large subunit, and then with respect to the small subunit. And then what this does is move the whole thing over by exactly 3 bases, so that you have the next codon can be read by the next tRNA. So the next tRNA is then brought in, and so on. And you can see how this happens. The tRNAs keep coming in and going through the ribosome, and see how the parts of the ribosome are moving. It's a highly dynamic machine. And the newly made protein comes out of this tunnel in the ribosome. And remember, I told you it contains within itself the information on how to fold up into a 3-dimensional structure. And so it folds up like that. And then this keeps on going until it reaches a special codon which signifies stop. There are 3 such codons of the 64 possibilities. And when that happens, a special protein comes in and it cleaves off the newly made protein from the ribosome so it can go off and do its thing. And then another pair of proteins come in and split the ribosome apart so that it can go off and start doing it again on a new messenger RNA and make another protein. And while you've been listening to me, the thousands and, you've got trillions of cells, and each of those cells has thousands of ribosomes, and they've all been churning away, making thousands and thousands of proteins in each of your trillions of cells. So thank you very much. Sure.
Questions? I am just the person moving the microphone around.
Guess that means you did a really good job.
One thing I was wondering about is how unique, so there's the question of the exact structure of the molecule that you infer from the crystallography. Is that structure unique? To what extent is it unique? And then there are these amazing dynamical processes you were just showing us. I mean, at what level is there uncertainty about any details, or is the case really closed?
So in crystallography, what you do is you trap a particular state of the molecule in the crystal. So depending on the crystallization condition, it has chosen a particular state of an ensemble of possible structures. And so you can think of it as a snapshot or as a sample of the ensemble. There are 2 ways of arriving at these dynamics. One is you can try and trap the ribosome at different points along that pathway. And there are biochemical tricks on how to trap it when a factor has arrived, when a factor has delivered the tRNA, when the tRNAs have moved, etc. So for 15 years, from 2000 to 2014 or 15, we would spend several years. Each structure would take several years to trap and then solve the structure of it. Since the development of direct electron detectors about 2011, 2012, and a lot of that work was done at the LMB where I work, it has now been possible to get high-resolution structures by electron microscopy. And because these detectors are so fast, you can do motion correction. So when you hit samples with an electron microscope, they get charged, and then they start moving around in the electric field because of the charge. But you can correct for the motion because the detectors are fast enough. And so with motion correction and with the greater sensitivity of these detectors, you can now get high-resolution structures without crystals. So if I had to solve a ribosome structure today, I would never bother crystallizing it. It would take a week from start to finish by electron microscopy. And so what you would, and the other advantage is that if you have a mixture of conformational states, you can computationally sort them out using electron microscopy. So you can get lots of structures simultaneously from one sample. So you can get an idea of the conformational variable.
That's what I was going to ask you about, basically, the role of electron diffraction at this point. So where's the field going? Is there more to be discovered about the ribosome itself, or are you do you move on to different things?
Well, I think, the basic structure of the ribosome is known, but now what and some of the mechanisms the broad mechanism of a bacterial ribosome is known. We don't have any idea of how a eukaryotic, a higher ribosome from higher organisms, including us, works in terms of knowing where to start on the message and how to start. And that's a process that's highly regulated and becomes deregulated in cancer and becomes hijacked by viruses, which have their own initiation mechanism. So they shut down the host initiation. So the field has moved on to more biological questions of regulation and control and sort of mechanistic questions. The basic structure is less interesting now.
So you talked about how you switched from physics to biology, but I was wondering how you became interested in physics in the first place.
Well, I think partly because you could say I was being a bit of a rebel. My parents were both biochemists, so no, I'm kidding. Actually, when I was in school, biology seemed incredibly boring because it seemed like a rote collection of facts that you had to memorize and regurgitate for exams. And physics, I liked mathematics a lot too. I almost decided to become a mathematician. And all of the adults in my life told me, oh, no, that's crazy. You'll never get a job. There are no jobs in mathematics. Of course, this was about 10 years before the computer revolution, but anyway, there we are. So I thought physics was like mathematics. It had the same beauty and logic to it, and that's what attracted me to it.
So I was just wondering,
You'll have to hold the microphone. Yeah.
Is this better?
Yeah.
Yeah, cool. So I was just wondering in your last video on the ribosome, just translating, so how fast does it occur, like the comments on kinetics?
So it varies. In bacteria that are growing fast, it can be about 20 amino acids per second. So you can think of the movie as about 5 times slowed down. Because if I showed you 20 amino acids per second, it would all just be a blur. But in higher organisms, it's slower. It's about 4 or 5 amino acids per second.
By gaining an in-depth understanding of the process, you've been able to make some impacts as far as developing antibiotics. What other major impacts have you recognized at this point of understanding?
I think other impacts are more likely to come later. For example, a protein is a particular kind of polymer. What if you wanted to make a completely different kind of polymer, but with different chemical properties? Well, maybe and you want it to be very precise length, precise order of building blocks and so on. So I think knowing the structure of the ribosome might make it possible to engineer so it can make unnatural proteins or it can make completely different type of polymers. And that's longer term work that's going on, in a few labs.
Thank you so much for your great presentation. My question here is the tRNA or other parts which are responsible for the reproduction of the amino acids are their motion is like a Brownian motion, or it's like they have a routine?
You mean their interaction with the ribosome?
When they come in and get out, is it a Brownian motion?
It's completely thermal motion. So the cell has 40 or 50 different types of tRNAs, and there's only one codon, and it has to select from those different tRNAs. And so which one it selects, is based on the matching. But it's constantly rejecting the incorrect ones which are coming in. So if I were to show a real movie, it would be incredibly messy. It'd be things coming in all the time and going out and so on, and less of a story like a Hollywood movie, sort of.
Thank you so much.
So you talked a little bit about using the structural techniques that you use to determine how different antibiotics bind to the ribosome and how that can be used. But what do you think is the logical next step in this field?
In the field of antibiotics? Or of the ribosomes?
In the ribosome field.
Well, as I mentioned to Ron, I think the field is looking at different types of ribosomes. For example, a few years ago, we solved the structure of the ribosomes from mitochondria, which we have, but all higher organisms have, which are completely diverged from our ribosomes and are the cause of many diseases, many genetic diseases mapped to mitochondrial ribosomes. The other thing I didn't mention, when you talked about practical applications, is that the regulation of translation is now shown to be important in lots of processes, including cancer and formation of memory and so on. So understanding how ribosomes and how translation is regulated is really a big fundamental problem in biology. So knowing how the ribosome interacts with factors and how the factors affect the rate of translation, that's another big question in the field.
Thank you.
So if you were my age.
I wish I was, but.
If I was, well, okay. I'm not talking about biology, I'm not talking about math, it can be anything. Where would you go? 19.
You mean in terms of science?
I mean in terms of anywhere. It can be science. I mean, would you, I don't know, right?
I think the two of the most exciting areas, I think today, are in artificial intelligence and in the brain. So neurobiology at a sort of high level, cognitive neuroscience and, machine learning and artificial intelligence. But that's very much a personal choice, I think, people should follow whatever it is they're interested in. I mean, one way, if you look at science, and I actually, just on campus today, I learned about, the real variety of things that are being done. And every one of those problems is interesting, So I don't think, I said artificial intelligence in the brain, but they're also two of the most fashionable areas, okay? They're two of the most crowded areas. So by the time you get to be good at it, they may not be that interesting anymore. You see what I'm saying? I mean, when I entered ribosomes, it was considered a dead field. People said, oh, we already know what ribosomes do. They translate and they have all these factors and so on. And as for the structure, that's never going to happen. So, the field has essentially done what it can and, people moved on. And even the early people who worked on ribosomes, Francis Crick and Sidney Brenner and all those people, they left the field, okay? They sort of said, time to move on. And so, and for 15 years it was unfashionable. And then suddenly, it became fashionable again. Now I have a sense, only the really hardcore ribosome people are going to stick to it, so.
Thank you.
You've talked about your career as a scientist, but you've also been president of the Royal Society, and that has given you a different kind of perspective about human problems, problems of scientists, and so forth and so on. Do you want to talk a little bit about that part of your career?
Okay. I did say in the book that I was kind of astonished to be elected president of the Royal Society, and I talk about the Matthew effect. This refers to, I'm not particularly religious, but it refers to Matthew chapter 13, verse 12, which is, "To him who hath, more shall be given, and he shall have even more abundance. And from him who hath not shall be taken away even that which he hath." Okay? So there you have it. So you win a Nobel Prize, suddenly everybody else wants to give you other honors. And I'm not saying that's why they chose me, but I can tell you, without the Nobel Prize, they wouldn't have given me 2 seconds consideration. Okay? So it was a kind of a surprise to be asked, and a privilege as well, and an honor. But it was also a challenge, because I had been a strict lab scientist all my life, okay? And I went to Britain relatively late in life. I didn't have a big network, political network. I was not a mover and shaker, head of big organizations, nothing. I'd never run a committee meeting before, okay? So suddenly, I was asked to do this, and I sort of foolishly said, okay, fine, I'll give it a shot, okay? And the thing so it's a steep learning curve, but what you do is you start thinking about science. When you're doing science, you sort of take everything for granted. You take it for granted that, there's a teaching assistantship, or there's a, facility, or there's the NSF, or, there's a national lab. These things didn't happen by accident, okay? Somebody in some room talked about, wouldn't it be nice to build an accelerator at Brookhaven, called for synchrotron radiation? Somebody else thought, wouldn't it be nice, Vannevar Bush, said, wouldn't it be nice, we should be funding science. It's the endless frontier, we've got to, fund science in a systematic way. So it all comes to people talking and thinking about science policy and science administration. Then you have other questions. How do you convince the public? How do you get the public interested in science? It's not convincing in the sense of being a snake oil merchant. You have to educate the public about why science is so interesting and also so important at the same time. And why should they know? Because they're paying for it, So that's another thing. Then you've got to get the public to distinguish between pseudoscience and science. Now with the internet, you can click on a web page. You have no idea if that person is talking sense or not. They all use scientific jargon. Negative energy. How many times have you heard negative energy? Negative energy has a particular meaning in physics, okay? But, there's no such thing, as the negative energy in the way they're talking about. So or homeopathy or things like that, which have no chemical basis at all. So, and then there are bigger questions like climate change and, vaccination, whether vaccination is, good or bad, and so on. So scientists have to reach out. So these are all things where independent, respected organizations, which are trusted, like the Royal Society, have a role to play. And then we also have a role to play in the conduct of science, in what is the right way to have a good science environment that encourages everybody to participate and feel welcome in science and so on. So those are things. And then convincing governments to fund science properly. Now we're in the middle of Brexit, how to keep our connections with European science. So it does take up a fair amount of time.
So at the end of the video, you talked about 3 different codons which can serve to terminate protein synthesis. So I was just wondering how it was determined which codons did this and how they do terminate the formation.
So there are 3 codons which are called stop codons. So UAA, UAG, and UGA are universally stop codons. So they're stop codons in essentially all kingdoms of life, except in mitochondria, UGA codes for tryptophan. But other than that, they're essentially universal. So, and they're read by a protein instead of a tRNA coming in. So there's no tRNA that recognizes them normally, okay?
So,
Yeah, you can.
you've already kind of hit the peak. You got one Nobel Prize. If you had to guess how you'd get a second one, do you have a new white whale that you're working on?
You know, at my age, I'm afraid I don't have any delusions of winning a second Nobel Prize. Okay, so I think people who won two Nobel Prizes usually won their first one fairly early on, you know, and so that's just how it is. You know.
Can we have a last couple of questions? Okay, one more. Yeah.
Oh, there's one right there.
What sort of differences or challenges did you experience in writing this memoir versus writing, manuscripts and producing literature?
So the first thing I decided was that if I wasn't going to be honest, there's no point in writing it. I didn't want to write a sanitized, how it was all lovey-dovey, everybody got along, and everything was very smooth, etc. But I also realized that if you're going to be honest, you also have to be honest about yourself. So I'm very quite open about all of the mistakes we made, including some real elementary blunders, that we made that cost us time, just to show that that's part of science too, that you do make mistakes. And also, choices, that I made, that some of them turned out to be okay, some of them were mistakes, essentially. So I talk about that. And I talk honestly about people, scientists as people. So I describe them, I describe a bit of their personality, sometimes I describe their appearance, or in some cases, clothes. I've deliberately not described women's clothes, okay? And partly because this book is slightly modeled after the double helix, but I didn't want to be, unnecessarily rude like Jim Watson. I'm very frank about the science, okay? So when I find somebody's made a scientific mistake or were stuck or didn't know what they were doing, I say that, okay? But I don't talk about their appearance except, so somebody told me you can't describe women as attractive, that's just a no-no. So whereas in my first draft, it was completely gender neutral, okay? But so now, I took out all the women's references. So now I say, and he was a really good-looking guy, and blah, 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 and so on. And so my son said, Dad, people are going to think you're coming out of the closet with this book. Anyway, so I think that's the sort it is. But it's a fine balance between being very frank and being unnecessarily obnoxious or rude. And I like to, so Richard Dawkins in his blurb describes me as a nice Jim Watson. So that's exactly where I am.